Thanks for inviting us here today. So we've had a trip from way up north to down here, and I must say I've enjoyed looking at the agricultural context of South Australia. Forget where I am, South Australia in the last week. So I'm Lynette Abbott. I've been working on soil biology for a long time. I've been working on soil biology before people thought it was worth working on, actually. But anyway, it's been good fun. So I'm at the University of Western Australia. And I'm retired now, but the team is still working in this area. So now, where do I point this? Does it work if I point it? It works, okay. So today I'm going to talk about plant-soil interactions. Am I in your way? Plant diversity, inputs, management practices, measuring soil biodiversity, and about the Soil Health app. And I'll start with the Soil Health app and I'll come back to it at the end if we get time. But that little logo is in the App Store, and there's an app for both kinds of phones, or iPads and other platforms. So it's a bunch of information about soil biology, and it's been funded by the National Landcare program that we set up. So it's a free app on the phone. Don't try to download it today. It's got a lot of downloading, but once it's on your phone, you can use it off. You don't have to connect to the internet. You can still look at bits of it without connecting to the internet. You can either connect to the internet or download it and watch it. So we'll come back to that as we go. So I've been doing research at UWA for a long time, and it's been funded by many agricultural groups. And in particular, when I started, I was funded by the Australian Wool Corporation, as it was called in the old days. And they were interested in arbuscular mycorrhizal fungi, of all things. So right back in the early days, how do organisms function in pasture systems? That's what I was looking at in soil. And of course, in those days, people did a lot of research on rhizobium, and that was very well established. But thinking about mycorrhizal fungi in those days wasn't common in Australia or anywhere much. And so I started in that space, but then broadened out into teaching and doing research on other components. But many groups, and probably your groups have funded me at some stage in my career. By the way, my career has been 49 years at UWA. So I'm packing together a long history of information and playing in this space. So it's a good perspective to look over time at this area. Now, these are some of the things I'm going to cover. So, soil diversity. I think when we talk about soil organisms, we really need to think about the soil that they're in, the soil type they're in, because although you've got lots of organisms and they're similar in different soils, but what's going on will depend on the soil. So if it's a heavy soil or a sandy soil, it's going to affect the habitat of the organisms that you've got. So the reason that is important is that if you're measuring soil biological processes or things, then what you get depends a bit on the soil. So you might get a different collection of data from a sandy soil to a heavy soil, but the sandy soil might be doing really well for a sandy soil. So just by numbers, it's not a good comparison if you're looking at cross soil types, which means on your own property if you've got a variation. But the soil-plant interactions with the microbes and the animals in the soil is really crucial. So you've got all these things working together, and to get a healthy soil, how do you manage that? How do you get that working? So soil contains mostly hard stuff that's not really biological, but the organic material, including the organisms that are in the soil, and even when they're dead, part of them, is a small fraction. And you think, oh, why worry about that? But that is the key to make soil work in any environment, including a natural environment. But in agriculture, that's the key. And we're focusing on that space today. Now, Colin will talk a lot to you about plant diversity. And the basis for me in relation to plant diversity is that having a diverse plant community influences the capacity of the soil to deliver a healthy soil. So if you just grow wheat in the soil, then you will have a limited soil biological contribution. You'll still get nutrient cycling, you'll still get all these things going on, but having just one plant over a long period of time will limit the capacity that these organisms can really contribute. Now, that's separate to thinking, well, you've got to manage a farming system, you've got to make money, that is all going on, but from the sense of having a soil that is going to work over the longer term, a variety of plant input is going to help that biological contribution. Now, there's a lot of organisms in soil. They do a lot of things. They work together, they eat each other. Whatever goes on in there is very complicated. And the picture on the left is close-up of a sandy soil. Even in a sandy soil, you've got hyphae connecting soil particles. Beside that is roots that contain fungi. These are the mycorrhizal fungi in roots. So these are in most plants that are growing in your soil. So the mycorrhizal fungi are a major part of the soil community, and they live in the plants, but they also extend out into the soil. Then roots, central. Roots are very central. We'll talk about that in more detail in a minute. You've got a lot of different groups of organisms that come and go in their cycling during the season. Rhizobia in the middle, key to getting good nitrogen fixation with legumes. So you've got most of our legumes that are in agriculture don't form nodules with the soil community of rhizobia. There are rhizobia in our natural ecosystems, but we bring in these agricultural plants, legumes from somewhere else, and then we have to make sure that we bring in the rhizobia, and that's been done very well in Australia over many years. Now, these little plates of algae, of bacteria and fungi, on the little circles there, growing, most organisms in soil can't grow in the lab. So most of these organisms that we talk about, we can't grow them. And in the past, we had sort of a vague idea of what they might be all doing, but now with the DNA technology, then we can have a better look at what is happening to the organisms that can't grow as well as those that can grow. Oh, by the way, just back on this one, scientists you have scientists who are experts on all these different bits in soil. So mycorrhizas, an expert, you have experts on soil animals, experts on different kinds of bacteria, but you have to manage all of it. So we get to experience knowledge about particular groups, but you are managing all of this complexity. Do you feel comfortable about doing that? Do you feel you need to know about it to do it? Well, you don't need to know everything about it to do it, because you've got an added advantage, because you know your land a lot better than the scientists do. And have you got a spade? Do you all have a spade? Yes? What do you do with a spade? Is it in the truck? Is it on the back of the motorbike or whatever? Do you just have a spade because you like having a spade? What do you do with it? You dig a post? Now, all right, so when you dig the hole for the post, do you look inside the hole? Oh, very good, yeah. Well, I'm thinking more of using the spade just to see how your soil's going, because actually along a fence line, it might be quite different to in the middle of the paddock. So just putting a spade into the soil can tell you a lot about the soil and about the biology. Now, the biology works with the plants, so it's not a separate entity, it's all combined, but to get a sense of your agricultural practices and how they're affecting soil biology, you can monitor this by just putting your spade in the soil, keeping an eye on how it's changing, because if your soil is like concrete, even in summer, it can get pretty dry here, so it might be harder, but even summer, winter comparisons, a spade is very helpful. Now, in the whole concept of soil health, soil biology, soil fertility, when I started doing my research in this area, I was interested in soil fertility. I thought, well, you know, you need a fertile soil to get things to grow, and there's a lot of literature being written on soil fertility. But as I was researching this in the early days, then soil fertility really just referred to nutrients. The main focus of research on soil fertility was, have you got enough nutrients for the plant to grow? And different plants have different requirements. So, you know, a lot of work's done in that space. And so soil fertility is also about the structure of the soil, and, you know, that you need it to be not like a rock. So there's a bit of the physical, but basically soil fertility was about nutrient supply. And then I'm teaching soil biology in the context of soil fertility, which is mostly about nutrient supply. And you've got a fertiliser bag, you can actually deal with it. So you've got the knowledge about the use of inputs is very well established, and biology has a role here, but you know, how do you get in? How does the biology get at the table, literally? Now these days we talk about chemical, biological, physical fertility. It rolls off the tongue, but if you then delve in and you say, I'm going to manage the chemistry, I'm going to manage the physics, I'm going to manage the biology, it just becomes a bit harder to explain what biological fertility is in this context. It sort of makes sense, we all talk about it now. But if you try to define it, you can handle the chemical side, and you can deal with it in relation to fertility for growing a particular crop. So that's sort of easy. You can deal with the physical. Well, you'll need a certain structure for roots to grow. The roots help the structure too, but you can get a handle on the definition there. But the biology, what do you need here for the biology? So actually, the biology is connected to the chemistry, because a lot of the nutrient cycling, that is about nutrients. The nutrient cycling is part of chemistry, but it's also part of biology. These organisms are cycling. So you don't need to just think about inputs of the chemistry, inputs of the nutrients, because you can take account of what the biology is doing. So that's a link. So you can't really separate the biology from the chemistry there. Then on the other side, the organisms also help in soil structure. The hyphae and the gums that are produced by bacteria as organic matter is broken down, that influences the physics. So in a way, you can't just keep them separate either. So you think it's great, we have chemical fertility, physical, biological, but actually when you try to dig down in there, they're also interrelated, that they can't be unpicked, which is a good thing. And hopefully by the end of our discussions today, we'll have some better handle on how we can manage this biology in relation to how we manage the other components of fertility. So if you're going to have a focus on the biological component, you can override that pretty much by adding a lot of fertiliser. So some of the nutrient-related processes that microbes contribute in soil, for example, can be overridden by just adding fertiliser and not taking account of an additional benefit from the organisms. So if you're going to have a bit of a focus on biological fertility, you have to focus on time, because it's not like putting on fertiliser. It takes time to develop. So in 1 year, you might get a build-up of my biological contribution to the fertility, but the next year and next year, gradually you'll get a benefit over time. So you've got to wait. And farmers I've talked to in the early days in Western Australia, when I was discussing this with them, they said something kicks in 3 to 5 years. They didn't know what kicked in, but they said something happened. So the question of time is not just a year, it's longer than that. And what happens is that the reliance on the biology then can be a contribution to nutrients in addition to the fertiliser by the cycling. Mind you, the cycling of nutrients occurs in the soil whether or not you put fertiliser on or not, because the organic matter is breaking down. That's a normal part of the process, but if you're relying a bit more on a biological base, then you take account of the contribution to nutrients for the plant. And the timing of the supply of nutrients will also be affected. So if you put a front-end loader of fertiliser on, or even at times during the season, you're getting a big burst of activity of nutrient supply, then that's very different to a gradual supply of nutrients during the season from the organisms. Now, you might say, well, I don't want to rely on the organisms because they won't deliver it fast enough to get the plants to grow properly. You know, these are all considerations and they're important. So if you just rely on biology, you think, well, am I going to get enough nutrients? But it's an issue to consider. So this has implications. So the implications for better reliance on a biological base, plant physiology. A plant which is getting nutrients gradually during time is going to have a different physiology than a plant that gets a big burst. And that may or may not be a problem, but it could be different for different plants, the extent that that matters. It can also affect grain quality, the supply of nutrients to the grain from during the season. And there's this matter of cost. And Colin's better at discussing this than me, but I say this could be financially okay, but I'm not a farmer, and we'll hear from Colin about whether or not he relies on this more and still is an active farmer, has the bank balance to match it, right? But I'm not talking about the money, but I'm saying you can still use a more biological base that can be profitable. So let's go back to this point about plant diversity being a key to building soil health in the longer term. What's going on in the soil? Now, this is a sandy soil, but the processes in a sandy soil are similar to a heavier soil. But a sandy soil can't hold as much carbon because the microbes, the carbon, organic matter is not protected, so the carbon is very accessible in a sandy soil, and so this becomes a problem if you want to build carbon. But remember, when you're deciding what your soil should be like, it depends on the soil type. So if you compare over the fence, it might not be relevant. You might have a very good, healthy sandy soil, and you've got to know what that might be, but a healthy sandy soil, a healthy heavier soil, a healthy acid soil, a healthy alkaline soil, they can all be healthy, but the numbers that link to those, the quality of those soils might be different. So just comparing soils without considering the soil type is not a useful thing. So, roots. The rhizosphere is the area around, or the volume of soil that's influenced by the roots. So roots are the bottom half of plants, and they get carbon from the top half of plants. The carbon, as carbohydrate, whatever, goes into the roots, and a lot of it just leaks out into the soil. So roots are very leaky. And so the nutrients that are in that and the sugars leak into soil, and that area around the soil is very nutrient rich or energy rich, and you get a lot more microbial activity around roots. Now, so does that matter? Well, you can grow plants in water where you don't have all this going on, but for a healthy soil, to get these benefits in the physical and the chemical fertility, then the development of these microbial communities around roots is very important. So you get a community of rhizosphere organisms, which is very diverse. They do many things and they rely on this carbon coming out of the plant. But within that community, you have pathogens. So pathogens, fungi and bacteria that cause disease of plants also occur in this space. And if everything's in balance, then the pathogens will just live in there as part of the community and won't get out of control. But if it's not in sync, then you will get disease. And that's another important part of managing the soil community. On the top right-hand corner, mycorrhizal fungi. Now these fungi occur in all plants except, which ones? brassicas, so different kind of mycorrhiza in the ericoid plants. So, you know, in agricultural plants, mostly they form arbuscular mycorrhizas, except for lupins, which is very unusual, why they don't form mycorrhizas, and canola. Now, in agriculture in Western Australia, we grow wheat, canola, occasionally lupins. You know, it's interesting, we're actually growing a pretty non-mycorrhizal system in our cropping systems in Western Australia. So the wheat is very mycorrhizal, but if you put a lot of phosphorus on the soil or even a lot of nitrogen, then you dilute out that fungal community. If you rotate with canola, you don't kill the fungi. These things don't kill fungi. What happens is that they just diminish in their abundance. So over time, if there's no roots to colonise, then you don't build up the numbers to carry over to the next plant. If you've got a pasture with a diverse community of plants, you'll have a diverse community of mycorrhizal fungi colonising those, and you'll maintain the numbers because you've got a lot of roots and you've got it continuous over time. When you put in a wheat crop, then you might decrease the mycorrhizas because of the high input of nutrients, but also because you've got less root matter compared to a heavy pasture. So you're thinking varying mycorrhizas is about varying root mass and varying nutrients inputs. So you can fiddle with the mycorrhizas quite easily by management practices. I'll talk about that a bit more later. Then you've got the rhizobial community, and if you're growing a legume like lupin, but also a lot of the pasture species, then you have to match the rhizobium to the plant. That technology is world-class, well understood. And I want to just highlight that at this point because there's a lot of interest in inoculating with other microbes in soil these days. And if you're going to think about buying something that is sold as a more general microbial inoculant, you need to go back to the rhizobium story, which is a gold star in terms of microbial inoculants. It's got terrific quality control. It's been studied for a long time. It's about matching, selecting, all of those things. Today we're talking about more generalist microbes, which don't fit that pattern so easily. It's not a one-to-one match between a bacterium and a plant, as is the rhizobium situation. And so we can't test everything. We can't test every microbe you put in. We can't test every mixture. We can't test every condition that you're going to use it. So we just can't know the level of benefit of microbial inoculants in the way we can with rhizobium. Just keep that in mind, and you're making decisions about what you put on the soil, and that's one consideration. Now, this is a poster developed by Gupta at CSIRO, and it was made a long time ago. He did some work with us, but this is something that he developed here in Adelaide. But it's a story, you won't be able to see all the details, but we can make it available to you. But it's about plants influencing microbial communities and soil animals. So you the plants grow, the residues go into the soil. They are a source of carbon and all other nutrients for fungi and bacteria. So they are quick colonisers of the plant material, whether they be roots or other residues on the surface. Then you kickstart all the animals. And this is like a zoo, but it's a pretty vicious zoo. These animals eat each other, they compete with one another, they don't respect one another at all. They just get on with dealing with the organic matter, interacting, releasing nutrients into the little red buckets. That's little poos, or that's whatever the dead organisms when they eat each other. And you build up this microbial base behind the scenes of this animal feast. And towards the end, you've got the bigger animals that come into the system. So the soil animals come in waves, and you look one day, you'll find some active, you look another day, different ones are active over time. But this is all going on, and it's not just the microbes that are doing the nutrient cycling. These guys are busy as part of that, and it's a massive community of action. Now, we did some work with Gupta in Western Australia, and this is sampling soil over the year, looking for the little micro fauna or mesofauna. Then the numbers, counting the numbers over time. In this case, we were adding different residues, so lupin and wheat residues to the soil. But the point is, if you sample out here, you think, I haven't got any of these animals, and then you go along a different time, and suddenly it rains, and then suddenly you've got all these animals there, and then later they've gone again. And that's just the cycle. So I think it's good in a general sense to understand, if you're managing these organisms in soil, you can't manage every individual organism, but understanding cycles, their life cycle. So they have, the animals have a very clear life cycle. They come and go, they disappear. The earthworms have a different life cycle. They can bury, come back up. The microbes have another life cycle, and often they're just inactive and then they're active, depending on their conditions. So we manage these organisms in relation to how we manage the soil. So if you grow certain plants, you'll help promote certain organisms to grow, and if you put fertiliser on, you'll do other things. You won't kill organisms by adding fertiliser. In fact, you can increase the numbers by adding more. The fertiliser will increase biomass of plants. You can have more microbes. It's not about killing things, it's about changing the environment, and that might just change the dominance of certain groups, and this may or may not be important. It's hard to predict those things. But everything you do will change these organisms in soil. Now, we've done some work on grazing. Now, grazing, how many people graze? Like, you have animals that... Who are cropping people? Some of you. So grazing is a really important part of, if you've got pastures, of course, if you've got animals, how you graze matters. And I'm not an expert in grazing, but I've got a pair of scissors, and we did an experiment using scissors. Now, scissors are not good replicates for animals, but you know, in experiments, sometimes you have to compromise. This was a ryegrass experiment where we were cutting the tops off the plants over time, over quite a long time, and to see what happened to the mycorrhizal fungi in these plants. This was a mycorrhizal study. We had a student who did this work who cut either, or didn't cut, or cut at different amounts and over time, different rates and degrees. So the impact of grazing, or cutting in this case, has huge impact on roots. So you cut the tops, you cut the bottom. You're not digging the soil up, but by cutting the tops off, you're influencing the roots. Not all plants respond in the same way to defoliation of the tops, but in this case, defoliation of the ryegrass reduces root growth massively. But what also happens is that it reduces the amount of sugars in those roots. So you've got more than one thing happening. So you've got less roots, and the roots that are there when they're heavily grazed have got less sugar. Sugar is all about microbial impacts, so these things are all connected, remember? So in this case, you see up the top, where there was no grazing, then over time the sugars built up in the roots of these plants, and when it was heavily grazed, you didn't have many roots, but there was a low concentration of sugars in those roots. So these impacts are going to affect the biology of the soil, but it's sort of important to think about the consequences of that. So if you're heavily grazing, you're going to be grazing the roots at the same time, and the roots are important in organic matter build up in the soil. So you're actually working both ways. So you all have your knowledge about how you graze, but this is a little extra thing to consider, that you're actually influencing the roots when you graze as well as the shoots. Now I'm going to talk a little bit about soil amendments, and this is something that we did with Mark Farrell, who's going to tell you about this. So Mark had a GRDC project, and we did some work with Mark about looking in the literature about all the work that's been done on soil amendments in relation to their effects on soil biology we were interested in, and funded by GRDC. And you're going to cover this at all in your talk, Mark? Okay. But you can ask Mark about the questions about this. But the point I just wanted to put here is that over the years, we've done quite a lot of work on soil amendments, and the reason that we've been interested in this is it's about, if you're going to stimulate microbial activity in soil, they have to have some carbon. And the question has been, is there an impact of adding compost, not across the whole wheat field or anything, but in more localised situations? What is the impact of these biological amendments on soil biology and how it might last over time? So this is a review. Now, the review was written, but you can't get access to that review unless you pay money. That's the nature of science. We lock up our science so people who want to know about it can't get access to it, except there's more journals being written now that have free access to journals. But I don't have you, Mark, yeah?
So Lynn McDonald did a plate English.
I've got it next, yeah. No, the exact point, yeah. But I wanted to make this point about science because a lot of, like this was funded by GRDC, but the review that was written, a scientific review then published in the scientific literature, goes behind a wall, and you can, if you're in a university, you can get access to it, but if you are just sitting at your home computer, you have to pay. Unless you know us, but that's another little point here. But, oh, sorry, I better talk about what's on the slide. So this review covers lots of different types of carbon-based materials, so biological inputs, and what the effects are. And there's the different inputs can have different impacts on soil biology, and they can have a short-term effect, a long-term effect. They can have different impacts on soil chemistry, soil physics. So you can't say any carbon product has a certain impact. There's a lot of variation. And a lot of these materials that people add to soil are very variable themselves. So even a compost can be different from here to there. So you can't say a compost does this. There's a lot more behind these things. But in this review, we looked at how soil amendments influenced chemical constraints, physical constraints, and biological constraints. And some of these inputs had impacts on all three or more than one. So there are complex interactions when you add soil biological amendments to soil. And in some situations that can be done, but in broadacre cropping, that's obviously less likely. So this is the version of this that Lynn McDonald wrote up for GRDC, and it's on their website, and you'll have access to the link to this. So this has been written up and it's accessible. So that's good because GRDC has made it available, and Lynn wrote it up. But the point about it is that information from science is not always available to you people who actually pay for it. Now, following on this, we did some work with the Southwest Catchments Council in southwestern WA. There are a lot of dairy farms down there, and they've got a lot of effluent, and they want to know how to deal with the manures and other things. So we did some studies. They set up 1 hectare plots of different treatments on the 3 dairy farms, and we went in and studied the microbiology of these situations. Now, I'm putting something very complicated in front of you, but let's start. So these days, we've got DNA technology. Have you got any DNA in you? Yeah, you got a bit, yeah. So all the organisms in soil have got a bit of DNA as well. So these days, we can go in and take a sample of soil, extract the DNA, and then we can get the sequences of the DNA all printed out, and it's a massive database. And I'm not an expert in this, other people who work with me do this. So you've got this massive, you've got lots of data about the sequences of the molecules here. Then you say, you can sort them because certain groups of organisms have certain patterns of DNA. Now they also have certain patterns that will be a blueprint for certain proteins and enzymes, which is really what they have the blueprint for anyway. So you can actually go through this database of DNA and identify how many genes are in this that will code for certain enzymes. In this case, that will break down certain bonds in different carbon molecules. So, long-winded. So down here, there's a list of enzymes that have been coded for in the DNA of this microbial community, bacteria in this case. So not worrying about who it is, but saying this whole collective of DNA. And then down here, there's a list of, next to it, that these enzymes can break down these molecules. So lignin is the toughest, and you've got certain enzymes you need to break down lignin. Now, so this is not saying who's there, it's saying what capacity this soil might have for breaking down these molecules. So it's a sort of it's not straightforward situation, but we can look at soils for their potential to do something, in other words, based on the DNA. And the pretty colours here, is anyone colourblind? No? Okay. So the patterns were the treatments. So these sites had manure added, compost added at 2 levels, and just fertiliser added. So this experiment was looking at the effects of these amendments in dairy soils, and in this case, in their capacity to break down carbon. That's not that related to probably what you're interested in, but this is the sort of thing we can do now, and we can make predictions about the capacity of that sort of function. But we don't need to know who is doing it. We can work it out without knowing who is in the soil. But notice that all the treatments had fertiliser added as well as the input, because at this point the farmers weren't really happy about leaving off the fertiliser. So the idea that the soil amendments might be a partial replacement for fertiliser, at this point we're not comfortable with that, so we add the fertiliser as well. But we can still study the effects, but later over time the decisions could be made to reduce the fertiliser, but at this point they weren't happy to do that. So that's what we studied. I'm not going to go into the details of the effects of that, but what I am going to say is that we did this at different times of the year. We did it in winter, which is this set of data, and we did it in summer, and the data are not the same. So while we can do this detailed explanation of something, it depends when you take the sample. And you can't take a sample of DNA every day and look at it. You can't do at that level. But you can understand maybe in different seasons you've got different processes going on, and you can take that into account. But just grabbing a bit of soil, doing a DNA test is not going to tell you the whole story about what's going on. And it's expensive. Now, in another study, one of our students investigated adding these things. So adding biochar, a biosolids product, and this was actually a lime biosolids clay mix. It was made by the Water Corporation in WA some years ago. I don't think they make it now. But the idea of this was to purify to reduce the problem with the pathogens in the biosolids. So, we had a mixture also of biochar with that product. So adding these amendments to soil, what happened? And we added these amendments to 3 soils, and they were pasture soils. And then this time, the blueprint of the DNA relates to who's there, the bacteria that are there. And in this case, all these names down here are different groups of bacteria. Lots of different groups. And you see that there 2 of them are dominant, 2 groups of bacteria. And there are some impacts of these soil amendments. I'm not going to go into the details of that here. But the point is, you can study this. You can study impacts of soil amendments on groups of bacteria. But unfortunately, within each of these bars of bacteria, there are lots of different kinds of these. So for the actinobacteria, there are lots of different groups within that single group. So the level of detail here can be, you look like you've got a lot of information, but actually you haven't got probably some of the important information. Because the other side of this is the length of the bar is the dominant, give an indication of dominance. But in some cases, you can have organisms that are very low in their abundance when you do this, but they have a very important impact. What am I trying to say? I'm saying you can get nice DNA patterns, but translating it in something that you can use to make a decision about management is like very long way. And so this is for understanding what's going on, but for you to make a decision, this sort of information, it's just not really, in one level it's very detailed, but in another level it is not dealing with some of the important information that you're going to need. But we can play around in this and get some understanding, and we like to do that. Staying with the DNA, this is some work we did with Dairy Australia, again in southwestern Australia. And we looked at some of these. Remember I said some of those bars had lots of different groups within them. So here's an example, actinobacteria, sorry about the missing A. So we took these samples from clover and grass, and we looked at the impact of nitrogen on the abundance of these groups. So this indicates this particular bacterium is common. This is a common one, but there are lots of other ones in this group, right? So when we added nitrogen or we grew a different plant, there wasn't much change in the community of this group of bacteria in the soil. There wasn't much change. There's probably changes, but at this point in time. Now, when we went to another group of bacteria, and if you start with the bluey bit up the top, you'll see that that has changed. The relative abundance of the groups within this group have been affected by nitrogen and by the plant. So, in other words, when you add something, a fertiliser or soil amendment, or you grow different plants, that affects some organisms in one way, other organisms in another way. So it's not a universal impact of management. Now, you don't need to know all this, but perhaps the reason for knowing that there is this complication is that someone can't tell you that if you do this, it will do something, it'll have this impact, because it's just too complicated. It's not easy to unpack it. And then remember what I said, if you sample in winter, summer, you get different answers. So this level of information is important for understanding what's going on and predicting what might happen. But at a management, for you to say, oh, I'll go and do this to my soil, or I'll go put this bug on or whatever, it's not that useful. Now, mycorrhizas, I have to talk a bit about mycorrhizas because I've spent a bit of time looking down microscopes. But the picture up the top is a mycorrhizal root. You can't see the fungi. The hyphae are spreading out into the soil associated with the root, but you can't see the fungi. So if you go and dig up your roots, they'll be full of these fungi, unless they're brassicas or whatever, but you can't see them until you cook them up, get rid of the cytoplasm, stain them with a stain that stains the fungus, and then look under the microscope. So the roots are full of fungi. They're plugging in to the carbon. They're not waiting for the carbon to leak out into the soil. They're just plugging in, helping themselves to carbon, and working very happily with the plant in either helping nutrient uptake from the soil, or the hyphae helping aggregation of soil particles, wrapping around particles, tying bits together, which is very important for protecting soil carbon or water, all of these things that are happening in soil. So they have a very wide benefit. But in fact, it's very hard to pick that benefit up scientifically in the field. We know they can do a lot of things. Now, these are three groups of fungi. These are whopping great spores. You can see them without a microscope, and they're the biggest of all fungi, and they live in the summer dry like this, and when it rains, they germinate and grow. They're happy in your soil. If you really want to know, if you give me a sample, I'll find these fungi in your soil. Now, similarly, in this study, we looked at soil amendments, so mineral fertiliser compared with a microbial inoculant, whole group of bugs, and a chemical fertiliser, and we said, how did these amendments affect mycorrhizas? So this is again a DNA study. We identified them using DNA. So we've got the 3 fungi, we've got the treatments with these different fertiliser or inoculant. We looked at the relative abundance in clover and ryegrass. So why am I showing you this? Firstly, to show that you've got these 3 groups of fungi and they're colonising the roots of both the clover and the ryegrass. So there's not specific. You don't have one mycorrhizal fungus that will go into a wheat plant, one that'll go into a clover. They'll go everywhere. They'll go into all plants. So the interesting thing is that plants are letting them in. Because if you've got a pathogen in the soil, most roots exclude the pathogens. So if you've got a tomato plant, it will be excluded by a fungus that colonises wheat roots. So there's a lot of specificity in rhizobium. You have to match the bacteria with the plant. And there's a lot with pathogens. The fungus or bacteria causing disease will be matched with the plant. But these guys will go into everyone. They all talk together. It's very interesting that there's no boundary. There are no blockages that stop this. So you've got them in your soil. So the more roots you grow in the plant, in the soil, the more of these fungi you'll have. It's just a given. So the best way to get them built up in the soil is to grow plants. Yeah. It was a mixed bacterial inoculant. It had a whole bunch of different things in it. Now, I'm going to not look at the details of the treatments in this case, but the point is that you do get a difference in the dominance of some of these fungi. You get more of these in this than in that, for example. And for this one, this treatment, the chemical fertiliser enabled some fungus to get into these roots, but not into these roots. So there was an impact of the chemical fertiliser on this fungus when it was growing in ryegrass, but not when it was growing in clover. So there's a bit of complication here in the relative abundance of the different fungi in the roots, and a little bit of an effect of the treatments. But basically, all the mycorrhizal fungi in your soil will colonise all of the different plants that are in there, with a little variation depending on the treatment or depending on the plant you're growing. And this is just saying that again, highlighting this, that the top fungus was unaffected by fertiliser in this case, but this one was affected by fertiliser. There was less in the ryegrass. Now, I can't say whether this is going to have an impact or not. I'm just saying that in general, the fungi will go into all the plants, but there will be some minor effects on individuals. Now, who has done soil tests on their soil for biology? Biology? Some biology soil tests? Good. So we started out doing this with a microscope looking at soils, and we started out a lot with the soil fauna because you can see it in a workshop. And we did a lot of work with Wheat Belt NRM, and this little booklet is available on the Wheat Belt NRM website, and I can give you the link to that. And we did this for farmers, and it's a bit of a story about soil testing and what you can do on your place. So you can send your soils away to someone else, but what can you do on your place with your spade, with a few bits and pieces? And you can even, in this book, you can look at the mycorrhizas in your roots. We've given you a little test to how you can do that. So it's not straightforward, it takes a bit of fiddle, but if you're interested in looking what you've got, instead of sending it to somebody else, then that's a possibility. But the question I've put up the top, can a soil biology test be used as an indicator of soil health? Okay. People talk about indicators. Everyone wants an indicator. What's a healthy soil? And I already said that a clay or a sandy soil might have different things in it, but one might have a lower level, but it might be more healthy. So you've got to put it as the context of that paddock, that soil type. But I have a little bit of a bugbear here, and that a lot of people talk about indicators when all they really mean is a measurement. So you can measure a lot of things in soil, but it might not indicate anything. So you get a measure, oh wow, I'm really excited because I've got a measure, but what does it indicate? And that's based on understanding. You've got to understand before you can know if it indicates anything. So an indicator implies that you can predict something from that measurement, but just a measurement, you've got to put it in the context of how you took the sample. So a measurement is not an indicator unless it indicates something. And then you're thinking it predicts into the future. And if we're talking about sustainable, resilient systems, we want to take samples which we know that if we keep doing this, we're going to get a longer term benefit. So that's the indicator story. Now, you can measure a lot of things in soil, biological things. You can measure a lot of other things too. So you can measure who is there. You can measure the organisms that are there. You can get this amount of those organisms. So you can do the rhizobium, you can check your nodules, you can see if they're pink or if they're white, you can know whether they're functioning, but you can actually do these counts, you can count the worms and you can look at roots of a mycorrhiza. So you can look at the animals, they'll change with time, but you can look, so you can get these things you're measuring something, right? And you can measure the amount of these. But you can also measure what they're doing. So you can measure respiration in soil. That's the response of all the organisms collectively doing that. So that's a measure that you can take. And you can measure enzyme activity. You can measure these potential gene functions I've been talking about, the capacity to have these enzyme benefits for breakdown or whatever. You can actually measure gene function. You can actually measure the breakdown using these molecular techniques as well. So what are you going to pick? Which one do you want to use? Who's going to pay for it? You know, when are you going to do it? So what are you going to sample? When? Where? How many samples will you take? How often will you take them? So a random sample of soil is not going to be an answer to too many questions. It'll answer a question or several. But because biology is living and it's changing and dynamic, so when you sample it, something might be there one day but not the next because you can't pick it up. So what to sample really depends on what the question is. And it's your question that has to be answered, not someone else's question. If you want to compare two practices or to compare two soils or farms or whatever. But I just put this in front of you whenever you are taking a sample, then these questions are there. So you might find that you take a sample at one time, you'll get a different answer if you take it at a different time, but you might be able to understand why that's the case. So then that's not a problem. But just this is a very variable situation. It's not like taking soil pH. You know, that can change, but these things are a lot more variable. So what do you want to take the samples for? What do you want to know? How do you want to use the information? I'm going to jump to something else now. How much time have I got? Okay, I'm going to raise this. This is just like something that's a bit off where we are. But everyone talks about soil biodiversity, right? And wouldn't it be wonderful to have more biodiversity than less biodiversity? Because, yeah, more must be better than less. Well, there's a lot of argument in the literature about this. And again, I said different soils, a sandy or a clay soil, they might have, so this sandy soil might have less biodiversity than this one. They've both got masses of soil biodiversity, absolutely masses. But, you know, this one, it'll have less because of just the structure of the soil. So, quickly, if you looked at this question in relation to how this much disturbance of the soil you made. Now, disturbance could be in that not digging it up. It could be that you've got more plants, different plant species, or you've got different rotations, which is a similar thing. And then if you think a sandy soil is a lower productive soil than a heavier soil. So how do these disturbances in relation to soil type influence biodiversity? And this is what I think happens. So if you've got a sandy soil, you don't really want to disturb it much because the maximum diversity, based on this theory, which I won't go into, the maximum diversity in a sandy soil you don't really want to muck around with it too much. Keep it as stable as possible. I'm not talking about low no-till versus ploughing here, but this is in the broader sense of how disturbed it is. But if you've got a very heavy soil, actually you might want to disturb it a little bit. I'm not saying dig it up and turn it upside down and all of that, but a heavier soil could get heavier and be less suitable for microbial. And so that, let's say disturbance is a tickle, not a great big. So a heavier soil might need a bit of disturbance to maintain the microbial diversity. Now, this is a hypothesis. We can argue about it, but I guess what I'm trying to say here is that these questions about more or less diversity are relative to the local situation. You can't generalise about them. But I think some of these questions now, there's room to explore here. Yes?
I was going to say, what do you mean by disturbance if you're not tearing a soil up?
Yeah. No, I think it's... I gave a talk recently in Western Australia, and there was a farmer from America doing a video in from there talking about, and the term he used was disruption. And I think that's a better term. But I didn't change the slide. But disruption I don't mean tillage really. Disruption means you change the plants or you have that, so you introduce that level of diversity. I mean, that's also good to have in this sort of soil, but we need to be a bit more careful with these soils because we can destroy that diversity more easily in a soil like this. So it's a really good question. Yeah.
That goes back to would you then consider the yeoman's plough a disturbance or a major disturbance?
Well,
I'm not...
no, look, I think any sort of ploughing is going to be a major disturbance, yeah. And I'm not saying that hire is a major disturbance either. The trouble is when you introduce an idea like this that you run into trouble. But I'm just saying that if you just leave everything always as it is, that might not be the best thing. Like I think a little bit of, even in the garden, in your lawn, occasionally you want to aerate it. Mark, yes?
I'm up from a hat in the ring here. So, obviously, long before we managed agricultural systems that were lots of animals, things like that turned over bits of the landscape over time. It's contentious these days, but the intermediate disturbance hypothesis Lynn would be aware of was a long-standing ecological idea that you've got the best of stuff somewhere in the middle between the extremes. It's contentious for a number of issues that hypothesis these days, but nonetheless, I think the broad ideas still hold, I think, because we've adopted no-till so long in Australia. We're getting to a point where we've got a different set of problems. They're nowhere near as bad as they were in the realm of recreational tillage. It's effectively what it got to at times. But there are occasions when there are some level of disturbance is actually the lesser of two evils. And I think that's by quite different levels of science as well. And the reason we're seeing these things in Australia and perhaps not seeing them elsewhere is just how long we've been doing it, so doing no-till so well.
Yeah, so I've introduced it. I've got a hypothesis here. There's a whole backstory to this. The person who I've based this on is called Michael Huston, who has really developed this theory of disturbance and ecological function in a way that's really amazing. And so anyone who wants to talk about that later. But I think in the context of our agriculture, when we started no-till, we said don't touch, but then later there's been an indication that some touching is required, some tickling up. And I'm putting that in the context of biodiversity of microbes. And everyone is arguing now about biodiversity. So I'm throwing this in the ring, and thank you for bringing the conversation. The more questions about this. But I think this is a bit of an academics argument, but it relates to actually practice. Yes? Sorry.
With heavy sandy soils, we get non-wetting. If we do nothing, it stays non-wetting.
Yes.
You lightly scratch it,
Yeah.
you can get penetration.
Yeah. The sand, we work with a lot of sandy soils that are non-wetting in Western Australia, and actually by this is it, I'm talking about in the pots. There's been a lot of work in the field in Western Australia with claying and all that, but if you've got a pot of soil and it's non-wetting, the best way to make it wetting is to mix it up. And the soil, and because the particles on the surface of the sand that cause that non-wetting can get disrupted, and then you'll break that bond, and then the water can it won't be hydrophobic. But so look, I've put a whole can of worms here in front of you with this disturbance, but I don't think that's a problem, because I think it makes us dig deeper in understanding. And I can tell you a story where early on, when I said some soils need some disturbance, and I'm talking a long time ago, that didn't go down well with the true blue no-till people, because no, can't disturb this. So, you know, disturbance is also about aeration and providing that habitat for microbes, distributing carbon, making it a bit more accessible than organic matter. So it's a big question. Now, just quickly. Most of these organisms are involved in carbon cycling in one way or another, but they're not all dependent on organic matter for their carbon. Some of them get carbon from carbon dioxide. So it's a complex issue. And we've fiddled on the University Farm in Western Australia. University of WA has a farm which is a commercial farm, and we do some research on it. And we've fixed up a little patch. We don't have a lot of salt, but this paddock was pretty crappy. And we introduced a mixed perennial system. We introduced some shrubs, and very quickly we've transformed this small patch into something decent. I'm sure a lot of you have done that, but that's something I just wanted to raise, that the University of Western Australia has this commercial farm which we are using for our research, and we've got a long-term soil carbon trial with different perennial mixes at the moment. That's happening. So just to finish off, building soil health, building the resilience of the system. What can you do? Well, you don't have actually a lot of options, and you've got to make money, but changing plant species, introducing diversity, rotations. I grew up in an era where rotations with pastures was a normal situation, and it's not now in Western Australia. But you can have inputs, the inputs from the plants, root exudates, but you can actually add other strategic inputs. And what is going to happen is that you're going to build, the basis of building resilience to me is that biological processes are going to be involved, and they influence nutrient supply, plant physiology, and soil structure. And these are added value to just getting the fertility based on chemical inputs. So the old fertility story, getting the nutrients right from the plant, we're getting a bit more than just getting the nutrients for the plant right. We're getting a benefit about a more broad-based definition of fertility. And the Soil Health app, I'll just finish on that one. So someone has downloaded this information from my head. So I've been working on this for a long time, and I had a colleague, Cheryl Rimmer, and she's a bit of a techo. She said, "You got to make a soil health app." Well, I wouldn't know how to do that. And she made the app, and it's there for you. I mentioned it earlier. But this is something that is information. It's not something you can put things onto the app, but you can, when you've got time on your hands, you can flick through and hopefully get some information and contact me if you've got any questions. So that's it. Thank you.